our next speaker is Layla Hirsh. Layla Hirsh is qualified as a Consitech researcher. She has a PhD in bioscience and biotechnology at the University of Padua, Italy. She's a computer engineer with a master in computer science and associate professor at Pontificia Universidad Católica de Perú since 2008. She collaborates currently with the European Bioinformatics Institute, EBI, and research labs in Argentina, Italy, Germany, France, and Spain. She focused her research on repetitive protein structures, and she developed a predictor for repeating units used to update repeats-DB. So welcome, Layla, and we will be very happy to hear your talk.
First, I want to thank the invitation and of course, recognizing all the work that everyone is doing in the organizing committee and everything. And for everyone in Europe that is actually taking the long night. Yeah. So today I will talk about the characterization of repeat proteins. As it was already mentioned, I work all my PhD in this subject and actually all the rest of my career since I finished my PhD. I work on repeat proteins. So some of you I know already know repeat proteins. They have this quality on having these multiple copies on the structure, and they are really important on the binding. See? They are known because they are important on general binding, and they are usually involved in diseases. So they have become a really nice and interesting subject. So there is something really particular in the repeat proteins, and is that we have some main and constant skeleton. And we have these spaces in the structure that actually can be really different. So this is a particular quality that actually would really interesting if we are thinking about designing. But there is a particularity on this kind of proteins. If we look at the sequence is not that conserved, it's really degenerated. So if we analyze the sequence, we won't see necessarily a repeat structure per se. But if we see as the structure, we will see that really the structure is conserved. As we can see in this image, we see how the sequence has a really low sequence identity at 0.2. And instead of that, if we go into the structure, we have a 75 of TM score. And actually we will see this similarity in the unit. And this is really nice. So we actually have this classification for repeat structures. We have all these patterns. We work, and I have been working in actually the class 3, the class 4, and the class 5, which actually in the structure is more well, it's easier to identify the structure. We have the elongated structures, the closed structures, and the bits on the strings. And of course, we have a sub classification where we see how this idea of elongated structures can be like known as also solenoids, alpha beta solenoids, beta solenoids, and so on. So we have this, probably all of you have been working with some of these structures in particular. So we look at the whole bunch, at the whole group. And we also realize that it's actually, we can find these repeat proteins in almost all the organism. which actually is really interesting because they are present in there. So could be a really nice subject to study. So if we talk about the characterization, in general, we can do some experimental determination, but for repeat proteins, it's really, really hard because it's really time consuming. And of course, if we try some prediction by homology, it won't work with repeat proteins because of their structure, their features. So prediction by homology is almost not feasible. But what we did was to think about this unit structure. We identify that there were these patterns inside the structure. If we go into the sequence, it was not possible to identify this, but if we go into the structure, yes, it was possible. So we started with a really small bunch of repeat units, structures, like little fragments of PDB, identifying the repeat unit in a repeat protein. And we create this library, and then using one algorithm, we actually identify these repeat units in the rest of the other proteins based on this pattern. So we use an iterative process in where we started with this known and manually curated repeat units and create this fragment, and then look for a similar pattern in one protein. Then we identify this repeat unit and doing an iterative process, cut this fragment and then look for something similar to that pattern. So in general, when we started here, we find, for example, no, this is an example we already tried. This is one main unit gathered from the library, and then we identify this master unit. Then we ignore completely the library because right now we have one of the repeat units from the structure. Then we cut this fragment, and then we go to look for that another piece, another fragment for this unit, and go on and go on. And in that way, we create really new knowledge. Because at first we found one unit that was similar to our manually curated data in the library. But then when we go into this similarity inside the protein, the protein chain, we actually got a really big library. So at first, when we had the library, we started with 900 proteins. With 900 units, yeah? And right now we have, well, more than 9,000. This is the last update of RepeatsDB2. Right now we are working on the next update, and for sure we have more units, which actually is more knowledge about the repeat unit. And this is really, really interesting because with more knowledge about the units, we can identify not only the new repeat units and the new proteins, and identify faster for sure the repeat proteins, but we can characterize what's in each of the units. And that's what is really, really interesting. But before, let me present you RepeatsDB2. This is a new version. As I am telling you, the new version will arrive in some month. We have here all the curation of each protein chain with all the unit repeat information. So this is a view of the page. We have new visualizers. We will have newer in the new version, and we can actually identify all the repeat pattern. As I told you, this is interesting, but more interesting is to see how they well, now they present a new structural classification. We identify some new patterns, which actually were included in this new database. And the new version will have a classification based by fault, which actually is really, really interesting and maybe more easier to identify the things you are looking for. But as I told you, the idea of having this more information is to start doing some new research. You know that to do all the EA research and testing, we need a lot of data. And at this point, we were not able to do all this because we had only 300 proteins, 900 repeat units. So now what we look at, sorry, is more information about the units. And we were able to identify these patterns in about the family, about the repeat unit, which actually is more or less what PFAM does. But the issue with PFAM is that was based on the sequence. And when you see the sequence in a repeat proteins, you don't see this pattern, or at least you don't see the complete pattern, not easily at least. So we did this analysis. We identify all the fragments, all the repeat fragments, and so this is the case for the alpha solenoid. We saw how they relate. We were based on the structure. So you see this bunch is a Tal effector. This protein is one that I actually love. It's the one, and actually the one, that has the repeat in the sequence and in the structure. You can see how the amino acids are conserved in there. Really, really big time. With the rest, instead, the conservation is not that big. So looking at the structure and thinking only on the structure, we can see that even if we have a repeat unit with the same pattern, I mean like alpha solenoid, loop, alpha solenoid, alpha helix, loop, alpha helix, or alpha helix, loop, beta, we can see how the twist and the curve will create a different protein structure. Sorry for that. This is, I don't know if I can stop this. Okay, so we look, sorry. Okay, I am not so sure if the view was messed. Okay, we are. You still are looking for the presentation? Yes, no? Yes?
Sorry. Yes. The presentation is fine.
Sorry.
You can continue, Layla.
Yeah, okay. So this different twist and this different curvature will give us a different protein structure. Will give us a different function. And this is really interesting because we have in here more data about the function at the end. So if we see at the sequence pattern, we will see that there are some positions where the amino acid has a really high conservation. And this is when the designing has a lot to gain. Because if I want to recreate a protein, I will know where and what amino acids I need to set and which one can be like whatever I want. And this actually, this kind of analysis based only on the structure information lead us also to have a better PFAM output. Because at the time that when we do the analysis and the creation of the profiles of hidden Markov model in PFAM, we can actually use the structure information to gather a better repeat family, a better hidden Markov model pattern. So we know about PFAM. PFAM is a really big database. It's really old, kind of. We have this paper. The new update is ongoing, but right now we have in here some of the last results. So we used to have these patterns, these hidden Markov models in PFAM that actually had the unit information, but if we use the structure information, we actually gather a better idea of this family, protein family. So before using this structure information, we got these protein families, this idea of this. In this case, we are looking at the case of the pumilio, but using the structure, the unit information is better. We don't have these gaps between the repeats units. The model is really more accurate, and we are mixing more or less on the process of the curation this structural information with actually a better result for sure. This is a manual process, but right now actually I am working on a tool to do this kind of automatically, but not 100%. For sure, this process of the curation in PFAM is a manually curated process that needs for sure someone to make the final adjustment. And of course, all these repeat proteins came with a big project that actually I must say, I think some of you have been part of the project. This is REFRACT, that actually is a repeat protein function and annotation and classification of repeat proteins. For sure is a really big project that includes not only Latin American labs, but also European. We have the EMBL-EBI, we have Sevilla, we have La UPO, we have Montpellier, CNRS, sorry, I remember that one in Italian, and others, which actually are part of this big consortium that is only working with repeat proteins. And we have for sure some more years on this research. With COVID, I think the years will be more. And that would be all from my side. But before finishing, I need to ask you one favor. I know you some of you are also students, so this is these are 3 forms that will help my student in his thesis. We are now also working in analyzing the usability of bioinformatic web services. I know we all suffer from this. As it was mentioned, I am informatician. My profession, I am an engineer in informatics. So right now I am doing also applying this in bioinformatics, and we are looking at the usability of these web services, which actually usually are a big problem in general, and we can get better with my student thesis. So please help us on this, and that's all. Thanks.
Thank you very much, Layla, for your talk. That was great. I think that Cristina Marino has a question for you. So Chris, do you want to unmute yourself and ask?
Thank you, Layla. Beautiful talk, amazing work. Thank you very much.
Thanks.
My question is more philosophical, which is, where is the limit between a motif or a repeat and a domain? For example, for some proteins it's quite obvious that it is repetitive, but I've seen, for example, a beta barrel, and I can think this the same an alpha bundle. At the end, which is the unit? Maybe a single secondary element. Another thing I have in mind, for example, fibronectin, that is 17 domains fibronectin. Is that a repeat or is a polydomain protein? Which is the limit there?
You know, we are still fighting around this. I must say, the project actually includes really nice people on the subject. We have Andrei Callava, actually for me is one of the best, if not the best in protein structures. We have Miguel Andrade, that is actually really good in sequence. In the project, we have all the really top authors that actually, well, the discussions in there are really interesting. But I must say, this is something we are looking for. Try to identify this, because some people always say one unit by its own is nothing. So you will need a second unit to do so. So what's the unit? Really a unit, or actually the two units to be
together.
This is something. Another discussion that is actually in there. Okay, what about the repeat units? We need 2 units to be a repeat, or we consider 3 units to be a repeat, which actually is also a big discussion in the database. And also because of the algorithm we use, we look for three units minimum. So this is one kind of rule that we set for the tool. Another discussion are the chains and the protein itself. The database is managed by chains. So we look inside a protein chain as an entry. But then you can see, and there are cases where if you see at the whole protein, you will see the repetition, but not in a chain. So this is something we are analyzing in all this research and all the secondments on the project. We have a lot of money just to make people coming from Europe and Latin America to Europe and vice versa, just to arrive to these conclusions, to decide what will be happening in the next few years. I think this is nice. For me, this is nice. I love the proteins because they look really nice. That's
yeah, that's true.
they are really amazing. I love them. But in general, I think the important issue here is that we are defining things. The project is to do so. As I told you at first, when I start with this subject, there were 300 proteins chains annotated by hand. Right now we have a lot of information, and it's actually really nice to see how they grow, how we know more. For sure, we do not know everything, but we are, I don't think getting closer is the right idea, but we are at least some fits more there. Like not for sure is a short path. It's really big, but this really nice. I think this is the nice part of bioinformatics, which actually I didn't have in informatics. This is changing, and each year appears something. And for me, the first thing I remember in my PhD was that someone said, like, no, repeat proteins is a repetition in sequence and in protein. And it was like, no, I see that it's not true. For TAL effector, yes. TAL effector, as I told you, is super particular, but it's one in a really big bunch of information. So the best and the biggest thing I remember in my PhD and the change for me was, I was an informatic, so my brain was 1 and 0, binary for sure. And now with nature, I know that this doesn't work. And this is nice. Everything changes. Natural, the war, well, right now is changing also. But nature is something that has no rules. We are trying to identify some, but in general, it's not binary, and this is nice. This is nice for us in the research, for sure. Yeah.
Thank you, Layla. Thank you very much.
Thank you.
So while we wait to see if anybody else in the audience has a question, I have one of my own, Layla. So it's a bit related to Christina's question, but I was wondering, when you search for these repeats within proteins, with which frequency can you identify parts of repeat proteins within other folds? Do you find that at all, or are they very exclusively always repeats? Or can you find them embedded in other folds sometimes?
Yes, we do. Right now we have, well, since I came back from Europe and I continue my teaching in the university, I have been working with my informatician students, sorry. So we have right now 2 new methods that allow us to identify repeat patterns using machine learning. And from this output, we actually identify some of these patterns inside proteins, but they are not repeats. So the classification measures the ranges we define where it's a repeat and not repeat. We are able to identify the repeat ones, but in the line, we can find these proteins that has some of this pattern, but looking at them, they are not repeats. And sometimes we find some new.
Thanks. So I think that we don't have more questions, so we're going to thank Layla. Thank you very much for your talk.